Dear students, today we continue our cycle of lecture of molecular biology, and today our lecture will be about cell cycle. Let's see it is our outline, and how you can see our first question is about cell cycle. We will be taught about interface, G1, S and G2 periods, and G0 stage. After that, we will talk about cell division, the different types of cell division such as mitosis, meiosis, antimitosis, polygamy, and amitosis. After that, we will talk about regulation of the cell cycle, about the checkpoints of the cell cycle. And after that, we will talk about the death of the cell, about the apoptosis and necrosis, and the main differences between them. So let's start. One of the main biological properties of the cell as an elementary life system is its ability to self-reproduce. Self-reproduction provides organism growth, development, and regeneration. So, cell cycle is the time between cell formation by the mother cell division and its own division or death. The mitotic cycle is the time between two cell divisions and all processes that occur during this time. In some cases, cell and mitotic cycle are the same term, but the cell cycle is wider. What associations do we have when we said cycle or cyclical with what geometric figure? I hope you said that it is a cycle. Okay. Look at the picture. Cycle. So mitotic cycle look like a circle. So often the mitotic cycle is illustrated by a circle like that. So this is a process that repeats cyclically through a series of consecutive stages. So the mitotic cycle of growing population will be divided to two big periods, and the first period is called interphase of the mitotic cycle. Interphase is the period between divisions when cell grows, perform its function, and gets prepared to divide. And another period of the mitotic cycle, it is the cell division or mitosis. Let's talk about the interphase in detail. The interphase may be divided to three periods. The first period is called G1 period. G it means growth or gap. The first period also called post-mitotic because it occurs after mitosis, or pre-synthetic period G1. In this period, cell just had division, so now cell will be growth, produce RNA, proteins, store energy, but they don't make DNA. In pre-synthetic period, cell nucleus contains diploid chromosome number. Each chromosome contains only one chromatid. Chromosomes are despiralized. After that occurs synthetic period or S period. The main event is DNA replication. DNA replication occurs. Each chromosome receives second chromatid. As a result, chromosome number is diploid. Each chromosome contains two chromatids. And after that occurs Post-synthetic or pre-mitotic period, period G2. So it happens after synthetic period, so it's called post-synthetic, and it is occur before the mitosis, so another name of this period is the pre-mitotic period. There is producing mitotic apparatus protein and producing and storing energy for fusion mitosis. And let's talk some words about G0 phase, or in another word, resting phase. G0 stage is the stage where cell neither dividing nor preparing to divide. In this period, cell actively perform its functions. Often cells are terminally differentiated and will carry out their function in the organism until they die. And after interphase, the cell division occurs. There are different types of cell division. Let's talk about it. The first type of cell division is called mitosis. Mitosis, or in another word, indirect division, is unique type of animal and plant cell division during which cells pass a range following changes leading to two-daughter cell formation with diploid chromosome number, which are necessary for all individual hereditary properties development. Another type of cell division is called meiosis. Meiosis is a special type of cell division that reduces the chromosome number by half, creating 4 haploid cells, each genetically distinct from the parent cell that gave rise to them. This process occurs in sexually reproducing single-celled and multicellular eukaryotes, including, as you know, animals, plants, and fungi. Another type of cell division is less common than mitosis and meiosis, and this type of cell division is called endomitosis. Endomitosis is the doubling of the chromosomes within a nucleus that does not divide. In other words, it is mitosis under the nuclear envelope. The doubling may be repeated a number of times in a single nucleus. This leads to the formation of polyploid cells. Look at the picture. What is polyploid cells? It occurs mainly in insects, salivary glands, and in some vertebrate tissue. Another type of cell division is called polyteny. Polyteny is the condition of chromosome, nucleus, or cell in which the DNA has repeatedly replicated without subsequently separating. After S period, cells go right away to the G1 period without G2 and M period. And let's talk about amitosis. Amitosis is a common type of the cell division. In another word, amitosis, named direct division. Amitosis is cell division by simple cleavage of the nucleus and division of cytoplasm without spindle formation or appearance of chromosome. Amitosis is typical for tissue where it is necessary to quickly restore the cell pool without worrying about the even distribution of genetic material. And now let's talk into detail about the mitosis and meiosis. As you know, mitosis is a process which includes four main stages. It is called prophase, metaphase, anaphase, and telophase. And let's talk about the prophase. Prophase, the main events of this period are DNA supercoils and chromosome condense, becoming visible under microscope. Let's remember our previous lecture. We said that the chromatin and chromosome, it is the same. It is a type of DNA, but we see chromosome under the microscope only in the cell division, for example, in the mitosis. So chromosomes will be in the cell division. Also, chromosomes are comprised of genetically identical sister chromatins joined at a centromere. I remind you that the centromere, it is a locus when the shoulder of the chromosomes are connected. This is the centromere of the chromosomes. Paired centromeres move to the opposite poles of the cell and form microtubule spindle fibers. So this is the centrosome. They are moved to the opposite poles of the cell and form microtubule spindle fibers. The nuclear membranes break down and the nucleus dissolve. The next stage is a metaphase. Microtubule spindle fibers from both centromeres connect to the centromere of each chromosome. Microtubule depolymerization causes spindle fibers to shorten in length and contract. This causes chromosomes to align along the center of the cell, equatorial plane or metaphase plate. This condition is called metaphase plate. Another stage, anaphase. Continued contraction of the spindle fibers causes genetically identical sister chromatins to separate. So the main events of the anaphase, it is the identical sister chromatins separates. Once the chromatids separate, they are each considered an individual chromosome in their own right. The genetically identical chromosomes move to the opposite poles of the cell. And the last stage is called telophase. Once the two chromosomes set arrive at the poles, spindle fibers dissolve. Chromosomes decondense, no longer visible under the light microscope. Nuclear membranes reform around each chromosome set. Cytokinesis occurs concurrently, splitting the cell into two. And now let's repeat all of our materials and see some funny movies.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached. And we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there, and it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Now, let's continue our lecture, and let's talk about meiosis in the detail too. An important difference between mitosis and meiosis is the presence of additional stage in prophase. So there are lectin, glucogen, actin, diplogen, and diakinesis. Lectin. In this stage, chromatin condensed into visible under the microscope chromosomes within the nucleus. Each chromosome contains 2 sister chromatids. Look at the picture, 2 sister chromatids, and each chromosome has homologue present in the nucleus. The homologue it is blue and red colors on the picture. During this stage, the synaptic complex begins to assemble. On the picture, synaptic complex, there is Figure 10. The chromosomes pair with their homologue forming homologous chromosome pairs, a process called synapsis. The paired chromosomes are called bivalent or tetrad chromosomes as they are formed by a complex with 4 chromatids. Bivalent, B, it means 2 in the Greek, and tetrad, another Greek word, tetra it means 4 in the Greek language. Pachygen, Crossover occur. crossing-over. Crossing-over is a process where non-sister chromatids of homologous chromosomes exchange segments over regions of homology. In the sites of genetic exchange, chiasma form. This exchange of DNA results in genetic recombination, the formation of new combinations of alleles or genes. Diplotin. Synaptonemal complex degrades, and homologous chromosomes dissociate slightly from each other, though they are still bound at the chiasmata, the regions of recombination. Diakinesis. Chromosomes fuse and condense. The chiasmata remain intact during this stage. The nucleoli disappear. The nuclear membrane disintegrates, and the meiotic spindle begins to form. And after that, after prophase 1 starts metaphase 1. Homologous chromosomes line up along the equator on the metaphase plate of the cell. Chromosomes pair are moved into position by the spindle fibers, which are not attached to the kinetochores at the centromeres. Kinetochores is part of the centromeres, and it is a complex of different proteins. After that starts anaphase 1. The spindle fibers shorten and the homologous chromosome pairs are separated from each other. This occurs as the chiasmata are severed, pulling homologous chromosomes apart. One chromosome from each pair moves toward one pole, with the other moving toward the other pole. Each chromosome still contains, I remind you, a pair of sister chromatids. Telophase 1. The spindle fiber disassembles. The nucleolus reforms. The genetic material briefly uncoils back into chromatin. Cytokinosis occurs, and after that starts interkinesis, or in another word, interphase 2, and in this interphase, no DNA replication. This interphase occurs without replication of DNA. It is important, please remember it. Meiosis 2. After meiosis 1, there are meiosis 2, and meiosis 2 includes division of the chromosomes similar to that of mitosis. This division separates the sister chromatids. Prophase 2. The nucleolus disappears and the nucleus breaks down. Nuclear membrane breaks down. The chromatin condenses into chromosomes. The spindle begins to reform as the centrioles move to opposite sides of the cell. Metaphase 2. The chromosomes each make out of two chromatids align along the equator of the cell to form the metaphase plate. In anaphase 2, sister chromatids are separated as the centromeres are clipped. The chromatids move to opposite poles of the cell. As the chromatids separate, each is known as a chromosome. Telophase 2. The nucleus reforms and the spindle fibers break down. The chromosomes uncoil into chromatin. Each cell undergoes cytokinesis, producing 4 haploid cells, each with an unique combination of genes or alleles and chromosomes. And now again we watch some movie to repeat all material that we taught before.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn-out cells. But not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis, you're going to divide twice and therefore do PMAT twice. And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now, I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs, and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other, and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different, though, from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line. They're in pairs in the middle. Now, during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have two newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm, but we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful of having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes, and the ending cells only had 23. So they are not identical to the original, and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Now let's talk about regulation of the cell cycle. How a cell understands to divide it or be in a dormant period. This is due to the regulation of some proteins. Siblings are among the most important core cell cycle regulators. Siblings are a group of related proteins, and there are 4 basic types found in humans and most other eukaryotes. G1 cyclins, G1S cyclins, S cyclins, and M cyclins. As the name suggests, each cyclin is associated with a particular phase, transition, or set of phases in the cell cycle and helps drive the events of that phase or period. For instance, M cyclin promotes the events of M phase such as nuclear envelope breakdown and chromosome condensation. The levels of the different cyclins vary considerably across the cell cycle. Look at this picture. A typical cyclin is present at a low level for most of the cycle, but increases strongly at the stage where it's needed. For example, M cyclin peaks dramatically as the transition from G2 to M phase. G1 cyclins are unusual in that they are needed for much of the cell cycle. In order to drive the cell cycle forward, a cyclin must activate or inactivate many target proteins inside of the cell. Cyclins drive the events of the cell cycle by partnering with a family of enzymes called the cyclin-depleted kinase. Abbreviation is CDKs. Alone CDK is inactive, but the binding of a cyclin activates it, making it a functional enzyme and allowing it to modify target protein. CDKs are kinases, enzymes that phosphorylate, it means attach phosphate groups to, specific target proteins. The attached phosphate group acts like a switch, making the target protein more or less active. When a cyclin attached to a CDK, it has two important effects. It activates the CDK as a kinase, but it also directs the CDK to a specific set of target proteins, one appropriate to the cell cycle period controlled by the cyclin. And there are the picture that's illustrated that we said. Cyclin activated CDK as a kinase. CDK phosphorylate attach the phosphate group to the specific target proteins. And the famous example of how cyclin and CDKs work together to control the cell cycle transitions is that of maturation promoting factor, or MPF in abbreviation. Like a typical cyclin, M-cyclin stays at low levels for much of the cell cycle, but builds up as the cell approaches the G2M transition. As M-cyclin accumulates, it binds to CDKs already present in the cell, forming complexes that are posed to trigger M phase. Once these complexes receive an additional signal, essentially an all-clear confirming that the cell's DNA is intact, they become active and set the events of M phase in motion. The MPF complexes add phosphate tags to several different proteins in the nuclear envelope, resulting in its breakdown, a key event of early M phase, and also activate targets that promote chromosome condensation and other M phase events. How does the another type of cyclin, APC, do its job? MPF also triggers its own destruction by activating the anaphase-promoted complexes of another named cyclosome, or in abbreviation APCC. Like a CDK, the APCC is an enzyme, but it has different type of function than a CDK. APCC causes M-cyclins to be destroyed, starting in anaphase, and causes destruction of the protein that holds the sister chromosomes together. Rather than attaching a phosphate group to its target, it adds a small protein tag called ubiquitin. When a target is touched with the ubiquitin, it is sent to the proteasome, which can be thrown off the recycle bin of the cell and destroyed. For example, the APCC attaches a ubiquitin tag to MC cleans, causing them to be chopped by the proteasome and allowing the newly formed daughter cells to enter G1 phase. So now let's look on this process on the picture. How does the APCC do its job? APCC adds a ubiquitin, the protein ubiquitin tag, to the protein called saccharin, sending it for recycling. Saccharin in normal binds to and inactivates a protein called separase. When saccharin is sent for recycling, separase becomes active and can do its job. And in normal activity, separase chops up the cohesin. Cohesin it is a type of substances which hold sister chromatids together. So sister chromatids separate, and in this moment of the time starts the anaphase. All cells move through the cell cycle. Do they breeze through from one phase to the next? If there are cancer cells, the answer might be right. It may be yes, it may be right. But normal cells, however, move through the cell cycle in a regulated way. They use information about their own internal state and cues from the environment around them to decide whether to proceed with the cell division. This regulation makes sure that the cells don't divide under unfavorable conditions, for instance when the DNA is damaged or when there isn't room for more cells in a tissue or organ. There are a number of checkpoints, but the three most important ones are: a checkpoint is a stage in the eukaryotic cell cycle at which the cell examines internal and external cues and decides whether or not to move forward with division. And the first checkpoint is the G1 checkpoint. It is located at the G1 to S transition. And the second point is the G2 checkpoint, and it is located at the G2 to N transition. And the last checkpoint is the spindle checkpoint at the transition from metaphase to anaphase. At G1 checkpoint, a cell checks whether internal and external conditions are right for division. Here are some of the factors a cell might assess. Look at the picture. Cell size. Is the cell large enough to divide? Nutrients. Does the cell have enough energy resource or available nutrients to divide? Growth factors or some molecular signals. Is the cell receiving positive cues such as growth factors from neighbors? DNA integrity. Is any of the DNA damaged? If errors or damage are detected, the cell will pause and the G2 checkpoint to allow for repairs. If the checkpoint mechanism detects problems with the DNA, the cell cycle is halted and the cell attempts to either complete DNA replication or repair the damaged DNA. If the damage is irreparable, the cell may undergo apoptosis or programmed cell death. This self-destruction mechanism ensures that damaged DNA is not passed on to daughter cells and is important in preventing cancer. M checkpoint, or in other words, spindle checkpoint. How does this checkpoint work? It seems that cells don't actually scan the metaphase plate to confirm that all of the chromosomes are there. Instead, they look for straggler chromosomes that are in the wrong place, flattening out, for example, in the cytoplasm. If a chromosome is misplaced, the cell will pause mitosis, allowing time for the spindle to capture the stray chromosome. You may think, or if it is a bad thing for cells in your body to die. In many cases that's true. It's not good for cells to die because of an injury, for example from a scrape or harmful chemical. However, it's also important that some cells of our bodies do die, not randomly, but in a carefully controlled way. Broadly speaking, there are two ways that cells die in multicellular organisms such as yourself. Necrosis is a process when cells are killed by things that harm them, such as toxic chemicals or physical injury. And apoptosis is a process when cells are triggered to undergo programmed cell death. Let's look at the main differences between the necrosis and apoptosis in this mechanism of pathogenesis. Necrosis. Plasma membrane is damaged. Cells swell up and ink contents leak out from the holes in plasma membrane and inflammation occur. Apoptosis. Apoptosis is the process without inflammation. Cells shrink and develop loops on its surface. The DNA gets chopped up into small pieces and some organelles break down into fragments. The entire cell splits up into small chunks, each enclosed in a package of membrane. And what happens to the chunks? They release signals that attract debris-eating or phagocytic immune cells such as macrophages. Also, the fragments of the dying cell display a lipid molecule called phosphatidylserine on their surface. Phosphatidylserine is usually hidden on the inside of the membrane, and when it is on the outside, it lets the phagocytes bind and eat the cell fragments. Apoptosis also plays a key role in the human development. For instance, as we saw in the introduction, your hand started out as a pedal-like block of tissue when you were an embryo. The block was carved into fingers by apoptosis of the cell in between the development fingers. Also, the main role of the apoptosis in the human organ is that the apoptosis can eliminate infected or cancerous cells. Apoptosis maintains the balance of the cells in the human body and is particularly important in the immune system. And now let's see some movie about programmed cell death.
Have you ever been sitting in class and thought to yourself, I wonder what my skin cells are doing right now at this very moment? This kind of pondering may be unique to me, maybe, but wouldn't we at some point wonder what our cells are doing right now? Because if you remember, as part of the cell theory, you are made of cells. All living things are made of one or more cells. Many multicellular organisms like you have cells that work together, working together as part of a body tissue, body tissues working together as part of an organ, organs working together as part of an organ system. Your cells are specialized to work in these different levels of organization. You have skin cells, stomach cells, muscle cells, just to name a few, and their functions need to be regulated. These cells actually are regulated as part of something called the cell cycle, and that is going to relate to my question of, I wonder what my cells are doing right now. Cells themselves can grow in size, but let's put it in perspective now. A multicellular organism isn't growing because each individual cell is getting bigger. A multicellular organism itself grows by making more cells, by the cells making more cells by dividing. That's cell reproduction. One reason that you are bigger than you were when you were 5, unless you are 5, is because your cells have divided to make more cells. Mitosis and the cytokinesis that follows that splits the cytoplasm allows you to make new body cells. But you don't want that cell division happening all the time. Why? It is likely that you've heard the term cancer before. We have had family members that have battled cancer before. It is definitely a relevant topic for all of us. Cancer is in part due to cells that divide too frequently. The cells are not regulated. They are uncontrolled. Cancer cells can have other problems too. They might not be able to communicate with other healthy cells. They may not be able to carry out normal cell functions. They may not securely anchor themselves like other cells do, which can make them more likely to travel somewhere else. Some cancer cells have the ability to secrete their own growth hormone that makes blood vessels divert over to those cancer cells and supply the cancer cell with nutrients, which can take nutrients away from healthy cells. Why do cancer cells become this way? Well, there's a lot of research in this area. With some cancers, there may be genetic links, making some cells more susceptible to having problems. These genetic factors might run in families. Exposure to toxins, radiation, excessive exposure to UV light, all of these can be risk factors for some cells to become cancerous. The uncontrolled growth that cancer cells have gives rise to more cells like them, which can develop into a tumor. Some tumors stay put, but some do not. Now, the good news is that scientists continue to develop better treatments, which include destroying the cancer cells with radiation or medication such as chemotherapy, which will target cells that divide frequently. Maybe someday you will be part of helping to meet the challenge of trying to eliminate cancer, because the fact remains that these cells are not participating in the cell cycle like they should. So, what is the cell cycle? The cell cycle is often represented as a pie chart like this. Cells are either in one of two different phases. A phase called interphase, where the cells themselves are growing, replicating their DNA, doing their cell functions, or they are in M phase, which includes mitosis and the actual splitting of the cytoplasm, cytokinesis. So this M phase is where cells actually divide to make more cells. But cells spend most of their time in interphase. So most of the time, they're not dividing. Now, depending on what kind of cell, it might do mitosis more or less often. For example, your hair follicle cells do mitosis frequently, which is why your hair can grow at the rate that it does. It's also why many cancer drugs may also target hair follicle cells, because many cancer drugs go after cells that do cell division frequently. It's a big deal for cells to hit this M phase. If a cell has an error, a harmful mutation, for example, you do not want it to divide, because then it will create another cell that has this same issue. That's where checkpoints come in handy. Along the cell cycle, there are checkpoints to check that the cell is growing well and replicating its DNA correctly and doing everything it's supposed to do correctly before it divides. To better understand those checkpoints, let's further divide this cell cycle pie chart. We have G1, S, G2, all three of those are part of interphase. Then we have M phase, where mitosis will happen. During G1, the cell individually itself grows. Then it replicates its DNA in S phase. You can remember that because the S is for synthesis, which means to make something, and it's making DNA. In G2, the cell grows some more in preparation for mitosis. So let's take a look at checkpoints. We've got one here in G1. This checkpoint checks, is the cell growing well enough? Is its DNA damaged? Because if it is, you definitely don't want it to move on to S phase where it would replicate DNA. Does the cell have the resources it needs if it were to keep moving on? This checkpoint in G2 checks if the DNA was replicated correctly back in S phase. Is it growing well enough? Does it have the resources it needs to continue? Okay then, moving on. This next checkpoint in M phase is my favorite checkpoint. It checks in the stage metaphase to make sure the chromosomes, which are made of DNA, are lined up in the middle correctly, that they're all attached to the spindle correctly, because if they're not, the chromosomes will not be separated correctly. So now you may have two big questions. First, what happens if the cell doesn't meet the requirements of the checkpoint? And second, what is doing the regulating of this cycle anyway? To address the first question, if the reason the cell can't go past the checkpoint is a reason that can be fixed, the cell may kind of pause here until it can fix that issue. But if it can't be fixed, then the cell does something called apoptosis, which basically means the cell self-destructs. This ensures that a cell that is damaged beyond repair will not go on to divide. So what is doing the regulating anyway? We've mentioned before that proteins are a big deal. Genes in your body can code for proteins that do an assortment of functions, and there are many proteins involved with regulating the cell cycle. Some of them are positive regulators because they allow moving forward in the cycle, and some are negative regulators that might make things stop. The proteins themselves can be sensitive to cues inside and outside of the cell. So, for example, two proteins that are involved in positive regulation are cyclin and CDK. CDK is specifically an enzyme protein, a fancy kind called a kinase, which is worth a Google. CDK can have different forms of cyclin protein bound to it. Different types of cyclin rise and fall throughout the cell cycle, and the rising and falling is based on a variety of signals to determine when the cell should move on to the next cell cycle phase. Typically, each cell cycle phase, G1, S, G2, M, will tend to have a different cyclin binding with the CDK. The rise and fall of cyclin types and the role CDK has when it's active is a fascinating subject to explore. Remember that vocabulary word we said, apoptosis? Proteins that are negative regulators, for example, a protein called P53, can be involved in initiating apoptosis. Again, we encourage you to explore beyond the video. One last thing to mention. There are some cells that don't go through the phases we mentioned because they're actually in G0. That's a 0, by the way, and not an O, because if it was an O, then it's a go, and G0 is kind of the opposite of that. G0 is a resting phase. Now, cells here are still performing cell functions, but they're not preparing to divide. Some cells go here temporarily, maybe if there's not enough resources around, for example. But some, like many types of neurons in your brain and spinal cord, may stay here permanently. If they stay here permanently, they'll never get to M phase, so they will not divide. This can be one reason why a major injury to the brain or spinal cord can have challenges with healing, as many of those cells may not be able to replicate. A topic that definitely continues to be researched. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So, today I will let you go. I would like to see you on the next week, and goodbye.